Good afternoon, ladies and gentlemen. My name is Claude Joly. I have been the Director of Electrode Research and Development for 20 years. I'm now doing more like expertise with electrode design. And I've been with MEDEL for 20 years. We've worked a lot on several drug delivery projects with European Union funded. And so now I would like to introduce you to cochlear implantation, an opportunity for introducing regenerative medicine, or regenerative biology meets the cochlear implants. It's the same thing. Yes, cochlear implantation provide a unique opportunity to introduce drugs or stem cells directly into the scala tympani. And what we would like to do maybe is regenerate deficient afferent inner hair cell synapse, IHC, inner hair cell synapses. We would like maybe to reconnect inner hair cells with unmyelinated afferent nerve fibers near the abenula perforata if it is retracted. We would like maybe to regrow inner hair cells if they are absent and reconnect those new grown hair cells with nerve fibers. And another way of looking at this is regrowth of myelinated afferent neurites towards the electrode to increase the neural density and proximity for electrical stimulation. So regenerate, reconnect, regrow. Here is a picture of what we are talking about. We are, with a cochlear implant, we are targeting the afferent nerve fibers. And the afferent nerve fibers might be connected towards the inner hair cells. Yeah, type 1 nerve fibers connected to inner hair cells, 30,000 type 1 neurons, 3,000 inner hair cells. That is a ratio that we have, and so we have many more like nerve fibers connecting to a single hair cells. It's almost like a 1 to 10 ratio. So yes, regenerative targets. What is it that we are trying to regenerate? We would like to try to regenerate the inner hair cells if it's missing, if a supporting cell is still present, and if we can send the right molecular signal for growing a supporting cell into a hair cell, and that could take place. We could reconnect the spiral ganglion cells peripheral process with a synaptic junction there at the base of the inner hair cells, and maybe regrow those peripheral processes that come from the soma of the spiral ganglion cells all the way to the hair cells. Yes, the correct neurotransmitter signaling must be established in regenerated sensory neural tissues. And what this means here is an inner hair cell with a single neurons doing a connection and a synaptic ribbon at the base where the action potential is being initiated through the glutamate receptors, neurotransmission. And so maybe it seems that in synaptopathy that maybe all what needs to be done on some patient might be to restore that neural signal through some nerve growth factors might even be sufficient to do that. Here is a study that was done a little while ago, not too long ago. This is temporal bone in the hearing population. So all of those are hearing people that had died and donated their temporal bone. And you can look on the graphs on the left, the graph on the left here. This is the total numbers of neurons starting of age 1 all the way to 80 or 90 years old. Those are all different cases. So spiral ganglion cells count between 30 and 40,000 when you start in life, and then you end up between 20 and 30,000. Yeah, and let's look at 5 of those patients. And let's see, here is 1, 2, 3, 4, 5 patients. And here on the right side of the graph, what we are looking is the type 1 fibers per inner hair cells. So how many synaptic connection ribbons is at different cochlear frequencies for different patients. And we can see that there is about 10 type 1 nerve fibers attaching to the inner hair cells. And it seems like the density is much higher in the speech frequency, speech reception region. And we can see there is between 15 synaptic ribbons, 15 synapses, 15 type 1 fibers connecting for each hair cells. But here is one patient in red that has, let's say, 1 or 2 connection per inner hair cells. So there is a great variation. Those were all people that died. Their temporal bone was examined, but they had no history of sensory neural hearing loss of any kind. So what it means is that there is a great variation in the number, there's a great redundancy also in the cochlea with the number of spiral ganglion cells and the synaptic connection. Here is another way of looking at that for the cochlear implant patient. Now we are looking at cochlear implant patient in that slide. We're looking at the total spiral ganglion cell count here on the y-axis versus the CNC scores, and all those black dots there are patients. And in that study, the group from Joe Nadol at Mass Eye and Ear in the USA found that there seems to be a correlation between spiral ganglion cells count and speech performance. Having said that, you also have to notice that with 2,000 cells, your score, your CNC score, which is a monosyllable score, can be anything between 10% or 30% or 55%. So it is not, there is a correlation. At the same time, we see such a variation in performance even with a low number of spiral ganglion cells. You have very good performers. So it's not a clear-cut correlation, and do we want to redraw spiral ganglion cells? Maybe it's not what we try to do. If you look at the morphological aspect of inner ear disease, what becomes very clear very quickly in those osmium black stained nerve fibers that you look here, you're looking at the spiral lamina of 3 different cases. 3 different cases, you're looking at the spiral lamina with a dendritic region. Let me see, maybe I can point this. Yeah, the spiral ganglion, this is a modulus. Okay, spiral ganglion cells will be there, then the type 1 nerve fibers and the type 2, they all branch out towards the organ of Corti situated here. But you can see a high density here of nerve fibers. You can see a thinning in that second picture here on the screen, and you can see even regions which are devoid of those peripheral neural processes. And maybe we would be interested in regrowing, regenerating, increasing the density of those nerve fibers. It is interesting to note that spiral ganglion cell survive as monopolar neurons. In normal hearing people, even in deaf patients, which haven't been deaf very long, sudden hearing loss, the spiral ganglion cells are bipolar neurons, which means that they have an axon, they have a cell body located in the modulus, and then they have a peripheral process that goes towards the organ of Corti and the hair cells. Long-term deafness causes retrograde degeneration of those peripheral processes, as shown in this picture. This was a high-tone hearing loss, a patient deaf for 50 years, and you can see that the neurons have actually turned into a monopolar neurons. Here is some of those monopolar neurons. So if they were bipolar, there will be another extension here of the dendrites going towards the organ of Corti. Here is a very interesting case which also can stimulate our thought in terms of what can we do on the pharmacological front with cochlear implants. This was a patient, a cochlear implant patient that died after 7 years implantation. He was implanted 7 years, he was an older gentleman. He lost residual hearing at implantation after 8 weeks. He was a residual hearing patient that got implanted with a very short electrode. A very, very short electrode. It was called the S8 hybrid implant. It goes no more than 8 millimeters inside the cochlea through a cochleostomy. And we can see in that preparation intense fibrous tissue growth in the scala tympani, in the scala vestibuli, and we also see a severe hydrops where the scala media is distended. You can see how the membrane collapsed, where the arrow is pointed to hydrops. This is the resonance membrane, and the resonance membrane collapsed during the preparation, but it was completely inflated by some, hydrops. So you look at this patient that lost residual hearing, and then you do a closer look at the inner hair cells count, at the outer hair cells count, at the spiral ganglion neural cells body count. And this is what you find. So inner hair cells count, where it says white, it means it's normal count. Where it's black, it's lacking. So we see that we have the basal turn of that patient, and so here is the electrode length. Okay, here is a frequency map of that patient according to Greenwood. This is a cochlear length. So no in black means 0, no. So no inner hair cell in the basal turn, but almost normal count in the apical region. No outer hair cell just about anywhere. This is typical of hearing loss. And then the spiral ganglion cells population, as you can see, is about 50% of normal. So this was amazing to see that despite loss of residual hearing, that the inner hair cell afferent nerve fibers were still present in that patient. Maybe it was just an issue of the synapse being paralyzed, being frozen, not being able to activate an action potential, according to acoustic stimulation. Or maybe it was a conductive loss due to the fibrous tissue growth. We don't know yet. Insertion of the electrode provides the first opportunity to bring drugs deep and along the scala tympani. So here is an electrode deeply inserted in a human cochlea. It's a representation, a graphical representation in a human preparation of a temporal bone with osmium stained nerve fibers that you can see. Intracochlear drug delivery in combination with cochlear implant is very possible. It's a very introductory phase for drug delivery to the cochlea for all kind of for correcting all kind of syndromes that patients may have. Intracochlear drug application in the context of cochlear implantation has the advantage that the cochlea is already open. because we're going to put an electrode carrier in there. In the future, intracochlear application could also play a role for indication other than hearing rehabilitation, since it appears to be necessary for certain therapeutic strategies, especially gene and cell-based therapy of the inner ear. This is a very nice article by Stefan Plomke. You can get this article and get more details about what's going on. Role of future biologically based therapy for CI. So I started the presentation with a description of the morphology of the nerve fibers and the status of those synapses and what can be regrown and what can be extended. And now I'm switching a little bit to the CI. In terms of the CI, what we can do, the goal will be as an entry point for drug delivery, stabilization of hearing loss, stopping its progression, preservation of neural integrity, improvements of neural responsiveness to electric and acoustic stimulation. That will be the first goal as a cochlear implant manufacturer, researcher, because we're going to go into the inner ear and we are going to intervene electrically there, but we can also intervene pharmaceutically to do some of those goals. Device. What kind of device we can provide as a cochlear implant manufacturer? We can provide combination product, doing both electrical and pharmacological stimulation, and we can also provide separate drug delivery system for the inner ear as a pre-intra-op function or even post-op. What have we learned from cochlear implants and hearing and structure preservation? We have learned that it is possible to open the inner ear through a round window membrane incision and preserve hearing. It is possible to explant and re-implant a cochlear implant and preserve residual hearing. We have cases like this. But hearing preservation levels are still too variable between patients. The same surgeon, the same ENT centers, the same procedure, and yet we have a variation of results, even though we are getting better and better all the time. There is still too much variety of results which preclude the entry into the cochlea just for drug delivery. Just for drug delivery, you cannot have any hearing loss or no more than 5 dB in pure tone average. We're not there yet. Here is an example of hearing preservation with long CI electrode. This is an article from Nordfalk in Hear and Hearing 2015. He had 18 Flex 28 and 17 Flex 31.5 patients, and you can see here their threshold at 125 post-op at 6 months. So there was a change in threshold. Each one of those dots are patients. We have Flex 28 patient, Flex 31 patient. 0 change of threshold on the y-axis. 0 dB means there has been no change. 10 dB means there's been an improvement in the patient pure tone average at 125 in this case. But this has also been measured at 125, 250, 500 hertz. So we can have better post-op performance. We have no threshold changes in some cases, but in numerous cases we have a threshold drop post-op, which can be on average 15 dBs, but sometimes it's 10, sometimes it's 5, sometimes it's 30, sometimes it's 40. And there is 3 patients here, as you can see, there is no threshold obtained at 125 hertz. Those are the patient with those little cross there at the bottom. It would be very interesting if we could introduce a drug during cochlear implantation that would bring all of those patients where those red dots are. That would be our first goal. Can we bring everybody around that 0 dB, maybe maximum 10 dB loss? Or can we even bring people with much better threshold with some kind of a drug intervention during cochlear implantation, during hearing preservation surgery. A new biological agent, therapeutic agent, facilitating such a goal is highly desirable. Once such a drug is discovered, direct access to the inner ear for drug delivery only may be secured. The objective of drug application in combination with cochlear implants would be the following: general reduction of insertion trauma, reduction of immune reaction, reduction of infection, reduction of loss of spiral ganglion cells, reduction of fibrosis and ossification, reduction of stimulation of non-auditory neural structures such as facial nerve, reduction of electrical channel interaction, and improvement of the frequency spectrum resolution and dynamic range of auditory neurons. Cross-sector interaction for intracochlear drug delivery. CI manufacturer can provide access to the CI patients for in-situ treatment, adaptation of the electrode and implant for short-term and long-term drug delivery as a combination device, and innovative development of generic devices and tools. For the academicians and for the pharmaceutical industry, drug discovery screening based on detailed knowledge of molecular makeup of disease would be important to flush out, and then further drug development optimization for intracochlear application, for incorporating with the silicone of our electrode, for example. So I will go through some example of some work that we have done with different academia, under European Union projects, multi-centers. Here we worked on a project where we actually looked at encapsulated cell technology. And what this is an implantable cytoreactor. Here is a cytoreactor. In the cytoreactor, we are putting some immortalized cell line that are designed to produce nerve growth factor. They can produce BDNF, they can produce GDNF, they can produce NT3. It depends how you want how you modify them. Those cells are encapsulated in a semi-permeable membrane, and that membrane lets the nutrient and the fluid somewhat come in. It protects the body from the immune reaction of a foreign body, such as cells, foreign cells, because those cells that produce those engineered cells, they were issued from a donor, from the retinal cells from a donor, and they were engineered and modified and immortalized. So you can encapsulate those cells, you can put a few of them in there, they can live for 1 year, 2 years, or even more. We can combine that with the electrode array, as you can see here. We've done some temporal bone study to be able to investigate this. So the temporal bone study was done in Hanover, that was also part of this project. Here is a cytoreactor. Here is the electrode. Again, a semi-permeable membrane lets the nutrient and some fluid in. It also let the nerve growth factors produced in that cytoreactors get out in the fluid and diffuse. Encapsulated cell therapy, it's a bit futuristic, but they are doing clinical trial for blindness right now. This company Neurotech, you can see their cytoreactor. It is placed in the retina. It is placed in the retina, it is attached with a suture right here, and its diameter is 1 millimeter, and the length 6 to 11 millimeters. We built kind of similar devices for the cochlea, but it was a much smaller dimension that we had to deal with. So as you can see here, NSGene is a company that was part of that European project called Know Your Ear. and we were trying to build an electrode that could actually take such a cytoreactor with it. Okay, so cytoreactors releasing BDNF or GDNF to protect auditory neuron, that has been tested in guinea pig. There was guinea pig studies that took place at the Karolinska Institute, and this material has been published. You can see the threshold the threshold of those guinea pigs that have been deafened. So there is an injection for neomycin at day 0, then a device plus a CI is implanted at 3 weeks, and you can see that the empty cytoreactor, meaning a cytoreactor with no immortalized cell line in there, you can see that the threshold shifts don't recover, they do not change, they get worse between 4, 5, 6 weeks, and then there is no more reaction. But the guinea pig treated with BDNF and GDNF shows a much lower threshold that seems to be stable, especially in the GDNF case. So histological analysis of those preparation is still going on. We also work with another project called NanoCI. NanoCI also had the aim of using nerve growth factors, BDNF, brain-derived neurotrophic factors, through a matrix inside the scala tympani, a matrix that would provide a scaffold for the neurons to project those dendritic process through the scala tympani, no more a fluid but a functionalized matrix, and they had some animal experiments which seems to have been successful. There was neuronal sprouting through that biofunctionalized matrix, a gel that had filled the scala tympani, providing a substrate on which spiral ganglion cells in those guinea pig preparation could grow their peripheral processes. Another interesting study, also with growth factor, has taken place in Japan. Japan have tried the use of IGF-1, insulin growth factor type 1, has been used in a clinical trial in humans, not with cochlear implantation. It was sensory neural sudden hearing loss patients not reacting to dexamethasone, to a standard of care, and they were treated with IGF-1 as a gel at the round window. So IGF-1 is a molecule which is available, which is certified, which is FDA approved for different applications, not for the ear, but for ALS and for also for people with deficiency of the liver or the insulin deficiency. Okay, but he had a protective effect. That IGF-1 has had a protective effect in those guinea pig study where they use IGF-1. They compare it with HGF, which is another compound, hepatocyte growth factor, and kind of look at in those guinea pigs preparation, it was injected in the ear, and they looked like there was a protective effect against after deafening the animal. As you can see on the right graph, a threshold. You can compare the gel with the IGF-1 or a gel with no IGF-1 or a gel with HGF or a combination of both, and it all seems to have had some beneficial aspect. So yes, molecules are available for doing some of those things. You can use a cytoreactor for nerve growth factors. They are the hardest things to deliver because they are proteins. They are proteins and they are unstable, and you cannot keep them long, so you need a cytoreactor or you need something very stable and a chemical species as like IGF-1. Gene therapy. Let me switch now to what else is going on with the inner ear in the context of cochlear implantation, perhaps in the future. Right now, there is a study using gene therapy, using adenovirus vectors for patients with severe to profound hearing loss, making a very small incision at the foot plate of the stapes and doing a very small, tiny injection of those adenovirus, which have been modified to alter the molecular signaling in the cochlea and regrow the hair cell. The goal is to have a hair cell regrowth trial. So those viral vectors are injected in the inner ear through a very small opening in the footplate of the stapes, a very small control injection. So things are progressing in an amazing way that we are actually doing a safety study on gene therapy for the inner ear now for hair cell regrowth, but it could be for something else. Instead of regrowing hair cell, it could be altering over molecular pathways depending on the condition. One other thing that we've done in the context of the Nano Ear project, again, European Union, thank you for sponsoring so many projects. Thank you, universities, for working with us on those projects. This was a design of a CI-based drug delivery system with a refillable reservoir. So you can see a cochlear implant here on the left picture, a cochlear implant, and here is an electrode. This is the electrode. He has a channel. He has a channel. And then here is a reservoir with two septum. I can inject the drug through that reservoir, prime the whole system, but I can also do a zero pressure injection to reload this thing. And you can see on the right side what that elution looks like in vitro. It can be used in combination with the cochlear implants, like shown on the left figure, but it could also be a standalone device if we knew how to enter the inner ear in a safe way without losing residual hearing. Then that could also be the next generation of device. Yes? We've also explored and are making available a single-use catheter for targeted drug delivery into the scala tympani immediately before electrode insertion in the context of cochlear implantation. So you see the catheter is at the same dimension as our electrode. It's a very atraumatic, very soft, very flexible devices. It's got a channel in the center. You can prime it with an insulin syringe of some kind, and then you can insert this into a scala tympani model, but you can insert it into a patient, into an animal. The drug delivery of the intracochlear catheter is designed to inject slowly 10 microliters of fluid in the scala tympani prior to electrode insertion. And it can be any kind of solution that does not have, which is a fluid, yeah? So here is some pictures of, in human temporal bones, we inserted that catheter, either 20 millimeters or 5 millimeters, and did a small 10 microliters injection, waited 60 seconds, 2.5 minutes, and you can see that that fluid that we injected diffuses very nicely within the cochlea, and it looks like a 20 millimeters insertion is a good way to go. And so now you know how much drug you put in that cochlea, you know how much, you know where, you know a little bit about the diffusion property of that drug, and you can start running clinical trial to see if it has patient's benefits. So we can make this a generic catheter. Temporal bone studies were made with this catheter where we injected a fluid with a contrast agent in the temporal bones. That's the two pictures you see on the right side. Temporal bone fluid injection. It's a contrast agent, a heavy iodine that you can see very nicely on CT scan. And you can see that there was no perforation and no fluid diffusion from scala tympani to scala vestibuli or scala media. It stayed in scala tympani, so that's a very good sign. This catheter is being used in one study in Hanover using stem cells treatment. So now we're switching to stem cells. We went to nerve growth factors. We went to gene therapy and adenovirus vectors, and now we are into the stem cells domain. So in this study, they are taking a patient, is going to get a cochlear implant. They extract some mononuclear cells from the bone marrow of the patient. They immediately process it, extract some stem cells, and inject this in the cochlea. This is what it kind of looks like: 10 milliliters of bone marrow, centrifugation, isolation, extraction of stem cells, and then injection into the inner ear with this catheter. So this is one usage for research which is taking place right now. It's nice. It can take all kind of drugs. This catheter. It can take stem cell. It can take other things. Finally, the final device that we are working, trying to secure that preservation of residual hearing during cochlear implantation, is a dexamethasone eluting electrode design, which you can see right here on that picture. You can see the contact 8, 7, 6, 5, 4, 3, 2, 1, and you can see a little band of white eluting dexamethasone eluting silicone there, a very small quantity, 16 microgram, which will elute for 6 to 8 months in the patient, suppressing inflammatory process, protecting residual hearing. This is our goal, and we've seen those results in animals very seriously. So we have a very good preclinical data so that we can go into a clinical trial with this entry-level device that could secure the access to the cochlea in a much more certain way. In summary, after this long presentation, thank you so much for listening, but CAI patients may have significant residual hearing in low frequency, even normal threshold. Electrode can be inserted, explanted, reinserted through the round window membrane with little damage, minimal loss of residual hearing, but the result in a patient is highly variable still. So in conjunction with cochlear implants, drug delivery could target specific hearing loss etiology with specific drugs, which we will not develop, but which we will collaborate with drug company or universities to bring into the patients, to bring into our electrode in one way or another. Not one drug fits all situation, except for preservation of residual hearing. That one drug should fit all patients because this is targeting the damage of opening up the cochlea, of inserting an electrode. But autosclerosis, cisplatin deafness, aminoglycoside-induced deafness, progressive hearing loss, sudden sensory neural hearing loss, they will all need their own drug, their own specific drug to correct whatever molecular mechanism is causing the hearing loss in those situations. Thank you very much for listening, and goodbye. Bye.